Yes, sir. We are seeing.
Yeah. So are they in slideshow mode? No?
Yes, sir.
Yeah. Shall I go ahead then?
Yes, sir. Sure, sure.
Yeah. Thank you. Thank you, bro.
Okay, sir.
So, well, good morning to all, and thanks for the Professor K. Samsarao Garu for his invitation to deliver a talk on agrochemicals and pharmaceuticals at Mangalayatan University, Jabalpur. So, just to make an aware, because I was told that the Department of Agriculture and Department of Pharmacy were there, the students of these two departments were there in this talk. So, I just want to illustrate what are the issues and pertinent to this agrochemicals and pharmaceuticals, and where we can build a career in respect to the designing the analytical strategies and understanding the regulatory aspects. So that is my institute, that is Institute of Pesticide Formulation Technology, which is established in May 1991 at Gurugram, Haryana state. So it's an autonomous institute under the administrative control of Department of Chemicals and Petrochemicals and Ministry of Chemicals and Fertilizers. So it is a 17 acres land allocated to IPFT by Government of India, which consists of three functional divisions. One is formulation development. So the second one is analytical division, and third one is the bioscience activities, what we have been doing it. So the main objectives of the institute is to develop and produce the environment-friendly pesticide formulation, user and environment-friendly pesticide formulation technologies, and promote the efficient application technologies of these formulations to meet the existing requirements even for the new formulations, and also disseminate the knowledge about the safe manufacturing practices, quality assurance aspects, and also to understand, to make an aware to the industry that raw material specification and source when they want to produce these formulations and pesticides at the larger scale, and also can do the analytical testing with respect to supporting the industry, and also with new and advanced analytical technologies, and also conduct skill development programs to make awareness among the farmers to safe and judicious usage of these pesticides, consumers and personnel involved in testing of pesticides, and continuing education programs. So we are in the process of establishing a PhD program at IPFT in the agrochemical sector, because this is the only institute in India which understand the requirement of agrochemicals and meet the requirements of those by producing the newer formulations as per the need and usage of these at the field level. So this is the role of IPFT when you think of about the pesticide, that is agrochemicals. So starting from the creation of the product through manufacturing process, and then application of this, and also the understanding the once we apply the pesticide onto the plant, so it will percolate into the environment through plant to and soil, and then water to the our food. So how to where this is going, so how this pesticide is occurring, and what is the fate of the pesticide once it is entered into the environment, and also how it is transported from these various sources like plant, soil, water into the food which we have been consuming regularly. So and then followed by the once we done the use of the pesticide, we have to mitigate it to protect the human health. So what could be the mitigation remediation? These are the four steps generally any product, whether it is agrochemical or a pharmaceuticals involves. So in which when we consider about the pesticides, IPFT has a role in during the supporting the manufacturing processes by the industry, and also developing the new formulations for application, and also doing the monitoring the studies to understand the occurrence, fate, and transport of these agrochemicals. And the one area which we are going to newly introduce is to develop the mitigation and remediation technologies. So that way, we have to produce the pesticide, use it effectively for the purpose which is required, then mitigate it to protect the environment and human health. So we are going to have an advanced research methodologies department, this R&D orientation, to develop the newer technologies to produce the mitigation and remediation technologies. So IPFT has a major role in agrochemical research. So we can develop, IPFT can develop so any type of formulation as per the requirement type and usage. So many times we may have to use emulsifiable concentration, suspension concentration, or like something like wettable granules, and even powder form, or in the concentrated emulsions, even in the micro emulsion form, and spreading formulations to arrest the larvae of the mosquitoes, the suspension emulsions, and gel and tablet formulation, and even if you can develop the formulation for the biopesticides, which is now the emerging area in our country. So far IPFT has developed 80 formulations and transferred to the industry, in which there have been many of them. Almost 60 technologies are in the market now. So we also has developed recently United Nations in the United Nations Industrial Development Organization, UNIDO. There is an organization which promotes the biopesticides alternative to the synthetic pops like DDT, because now DDT is eradicated and which is banned, and the production is also curtailed to a minimum level. So by I think 2025 we have to completely close the manufacturing process of DDT because of its toxicity issues related to it and its persistence to many decades. If you spray pesticide, DDT pesticide, and it will not degrade easily by the microorganisms available in the soil and all. So for which we need to have to monitor them, because and it will persist for many decades. So just to avoid those problems, now Government of India has initiated to eradicate it, and UNIDO and Government of India has now brought into develop the biopesticides alternative to the DDTs. So we have developed eight product formulations now. What is neem oil, mosquito repellent cream, spreading oil, botanical tablets, mosquito attractant trap, surface spreading. So all this now transferred to Hindustan Insecticide Limited. There have been now, we also have demonstrated the pilot scale demonstration, 2000 kg scale demonstration of these formulations to HAL. So now they are going to build it manufacturing in the metric tons production by 2024. And IPFT has an analytical chemistry division, which is having a world-class sophisticated instruments. We have 500 megahertz NMR and LC HRM, high resolution mass spectrometer like QTOF MS, and many GCMSs and LC triple quad MSs. And we have all the suspension stabilizer, emulsion stabilizer instruments for the formulation activity. So analytical chemistry division always supports the regulatory requirements of the industry for the for the registration of the pesticides. They can generate an analytical test report as per the CABRC requirement, Central Insecticide Board Registration Committee requirement, and can do the studies related to accelerated storage stability, shelf life, and container content compatibility test, identification and quantification of impurities in the agrochemicals, establishment of chemical equivalents, persistence studies in soil, water, and plant to understand their occurrence, fate, and transport, and residue studies during the pesticide application on the plant during the bioefficacy studies, and also pesticide residue monitoring. So far the analytical division of IPFT has completed 100 industry sponsored projects till date. So we also, the IPFT has enabled accreditation as per ISO IEC 17025, which is a regulatory requirement for the testing activity of the pesticides in various commodities. So we have a accreditation for 145 pesticide formulations and 278 pesticide monitoring in food commodities, and also 17 trace elements in food commodities. Apart from that, we also have the enable accreditation to monitor these pesticide residue and trace elements in water, that is drinking, surface, and groundwater. And also we have been participating in the Organization for Prohibition of Chemical Weapons, OPCW, the Netherlands proficiency testing program, which they will conduct yearly twice. So we have a CWC chemical and their degradation product facility to do that. And the IPFT is also certified by NGCMA, National GLP Compliance Monitoring Authority, for the GLP certified lab for the agrochemicals with respect to physical chemical testing, 5-batch analysis, and residue studies. So this is the this is the only government laboratory having a GLP certified facility for agrochemicals. So the IPFT, the bioscience division, which can do the field studies. We have some 8 acres of dedicated land to perform the field studies for the bioefficacy of agrochemicals, phytotoxicity, compatibility studies, and also to understand the effect of pesticides on natural enemies of pests, and pesticide persistent and residue studies. So we have been continuously supporting this bioefficacy and phytotoxicity studies for agrochemicals and formulations produced by the agrochemical industry. So we also have facility to do the studies of drone-based pesticide spraying and their efficacy on the plant, and also their and their toxicity on the plant. So which we have been conducting regularly. So the advantage of having a drone is that we can minimize the usage of the water during the pesticide application spraying. and also we can able to quickly spray the pesticides into the field. So it will take just 5 to 7 minutes to spray the pesticide for 1 acre of the land. So we also conduct the farmer awareness programs to make them aware about the safe and judicious use of the pesticides, because many synthetic pesticides, what happens, although there are instructions written on the bottles of these agro formulations when they buy from the shops, but they used to put it more than the based on requirement, which will be creating an issue with respect to the environment and human health. So just to make them aware, so what could be that, how to use it, how to protect ourselves when we spray the pesticide, and also what are the alternatives available, and also to do the zero budget farming. That means the local available plants, how to make an extract from that plants at the field itself and spray immediately, which will enable them to protect the crop in an immediate manner. So yesterday and day before yesterday, we conducted 2 programs in Haryana state. So we are every year we conduct 20 programs in every state of the in India. So we also do conduct the industry interaction meet. So to interact with the industry and understand their requirements and meet their and make them awareness about the regulatory requirements need to be produced during the agrochemicals production. So recently we conducted one at Hyderabad in association with NABL and the CABRC and IICT as a local partner. So this is all about our institute. So just now we will come to our topic, today's topic, agrochemicals and pharmaceuticals. So these both are become essential now in the today's scenario to protect the plant and human health. So the agrochemicals and pharmaceuticals play an important role due to their ability to prevent, cure the plant and human health, and helps to plant and humans for the healthy growth, and provides the food security and minimizes the malnutrition, and also provides the healthy life to the humans. So that is the most important. These agrochemical and pharmaceuticals are now almost going into the hand in hand with respect to their advantages, with respect to their disadvantages. So we will see what are their advantages and disadvantages in the next coming slides, and also need to use them judiciously as per the need and application to protect the environment and human health and to avoid the risk to the human health. So when you think of agrochemicals is a broad spectrum of chemical products comprised of fertilizers, plant protection chemicals, pesticides, and plant growth hormones used in agriculture. So these are the basic classes of the compounds. And nowadays there is a new chemical entities of combination molecules have been coming. So those also they've been depending on based on the requirement and the need and type of usage. They have been either classified into agrochemicals or into the pharmaceuticals. Pharmaceuticals, known as medicines or drugs, which are specially prepared in modern and traditional medicine. So they are essential for the prevention and treatment of diseases and protection of the public health. So this is the basic and broad definitions of these agrochemicals and pharmaceuticals. When we think of agrochemical and pharmaceutical, there are some molecules which are very common, but having both the properties to kill the pest and also arrest the disease. So this is something like, one example is boric acid. Boric acid is something like a jack of all trades. It has properties to kill the insects. So it has been registered in EPA as an insecticide to kill the cockroach, rats, and flies. And also it has been registered in the pharmaceutical sector as a ear drops and other medicines. Likewise, there is one molecule, chloramphenicol, which has been used as an antimicrobial drug for the pharmaceutical purposes, and the derivatives has shown the insecticidal activity and been registered as a pesticide formulation. So there are cases where we can found the commonalities, and the only difference is that the chemistry what we have been using. So the chemistry plays a major role in understanding what type of structure we can able to develop to move for the target issue. So whether it is to be used to cure the disease or prevent the disease, or whether we do use to kill the insect to protect the plant health. So when you think of agrochemicals and pharmaceuticals, so the advantage and disadvantage is agrochemicals. With respect to the agrochemicals, we can see it can produce the higher crop yield and increase the soil fertility, improved food grain production, vector-borne disease control, insect and pest management properties have been available, reduced losses from weeds, diseases, and insect pests. So when you think of the disadvantages of agrochemicals, environment pollution could be a major concern in recent times and decreases soil quality and nutrient imbalance. If you are using synthetic pesticide regularly, which is the same product, if you are using regularly for more than three crops, it can imbalance the soil fertility, and also it can decrease the nutrient availability to the plant from the soil. And contamination of the groundwater is a major concern, and toxic to soil microorganisms alter food quality, and residues effects on the human health. So this has been majorly been understood since many decades, and now I think we have been doing a lot of monitoring studies to understand their presence and what is the impact and the risk associated with it. And apart from that, also we have been trying to develop a mitigation strategies to minimize them in the soil, in the environment, and the human samples. Further, if it is something like a pharmaceuticals, and to avoid that, there have been now people have been developing like biofertilizers and biopesticides, and which are useful to microbes, and also organic pesticides can be biodegradable, so there will not be harm to environment and human health. So likewise, nowadays people are talking about personal pharmaceuticals and personal care products presence in the environment, which may impact the human health. Because suppose if you think of antibiotics, so there is now a lot of discussion about the antimicrobial resistance into the among the humans, because if the bacteria is able to induce resistance to the antibiotic what we have been using, so even when we are continuously consuming maybe 5 days or 7 days, if the bacteria has not been killed, so that will be a big threat. And many times the used strips or unused antibiotic pills, we just simply throw into the dustbin or somewhere in the sink or somewhere like. So those enter into the environment, so which may create a harmful effect. So now the regulators also now focusing on these uh, pharmaceuticals as an emerging contaminants, and trying to make the database of their availability in the environment to design a regulatory policy decisions to arrest them, because they may directly enter into the food chain slowly, so which is also not good for the humans. So when you think of a bio-based agrochemicals, to avoid the disadvantage of agrochemicals, what we studied in the last slide, so there are many factors in which now people have been promoting this bio-based agrochemicals because of their broad activity spectra. Bio-based agrochemicals means we are simply extracting either from the plant or from the some marine organisms or from the soil organisms. So when we just extract them, so it is not a single compound; it is a multiple compounds present in that extract. So for which we may develop the. So because of that presence of many compounds in the same formulation or single extract or single formulation, which may give a broad activity spectra, consistent field performance can be available, and a low cost product, because it is even some many times we can able to design a strategy to make them in the field itself. And then reframing registration and regulatory policies, because we may not much to be worried about the regulatory frameworks and requirements for these botanical pesticides. And awareness among the end users is very easy, because nowadays even villages, if you go, you can simply say that about what is the neem. Do you know about the neem? So everybody knows that it has some medicinal and pesticide agro based pesticide properties, insecticide properties. So that is very easy to make the awareness into the farmers, and ease of availability. Now we have because India is a naturally, its a biodiversity is very high with respect to the plants, so we can able to get that new botanical pesticides in a very easily for the agriculture or even for the pharmaceutical applications, and a longer shelf life. So there is a chance that if you can develop a effective formulation which can able to enhance the shelf life of this formulation, which is easy to use, and a better delivery system. So these are the molecules which have been having bioavailability and easily penetrate into the plant very well. So that may give very good advantage. Likewise, if you think of the herbal medicines, which has been now people have been talking about phytopharmaceuticals, then nutraceuticals, and all these things, we are now seeing there are many companies exclusively producing these plant-based herbal medicines for many to prevent or cure the diseases. So those are by category we can say that herbal pharmaceuticals, herbal functional foods, herbal dietary supplements, and herbal beauty products. So these are the sources from leaves, roots, and barks, fruits, whole plants, and others. So there now people are making these tablets, powders, extract, syrups, even extract being into a capsule form. So that is something like been lot of work is going on, and the CAGR is now going to enhance this tremendously about this phytomedicines. We are expecting to reach up to 411 billion US dollars, 411 billion US dollars of this market herbal medicine. So that's why these have been now promoted by the many organizations. But there are the issues also when we think of this biobotanical pesticides or herbal medicines. So because of the issues related to the herb and drug interactions, unknown composition. Because when we think of a single extract, it may contain the many hundreds of the metabolites in that extract. So identification of those extracts, many times people have been suspecting about the availability of toxic heavy metals in these herbal formulations. So regulators now brought that if you want to produce a herbal formulation, you should avoid to you have to see the presence of toxic heavy metals like lead, cadmium, arsenic, mercury, and all. And a lack of standardization for the dose of active component. So many times what this is extract, if you extract one time, we may get 100 compounds. If you extract the second time, we may get 90 compounds. If you extract another time, we may get 150 compounds. So that standardization of the standardization of the procedure to extract and also fixing the dose is becoming a little difficult. So that is the one issue now we have to address to stabilize the formulations for the herbal medicines. And your dosing is inappropriate, and pharmaceutical form, bringing into the pharmaceutical form, it takes little time because it is still in the standardization phase for many of the extracts. And there may be contamination because these are plant based. So since the plant is growing in the soil, so we may spray the pesticides, agrochemicals for to grow them. So presence of pesticides may not be good in the phytomedicines. So that is also one possibility of we have to check the formulation for the not only when we identifying the metabolites of that extract, but also to see the xenobiotics like agrochemicals presence in this extract before being used for the medicinal purposes. And improper labeling is one of the problems. So these are some issues which we can easily address with the development of the and use of the effective analytical strategies. Then after that, once we use these agrochemicals and pharmaceuticals, then we have to understand what is the fate of those and their occurrence, where they can occur and how they transport. So when we think of the pesticides, when we spray it will, onto the plant, so it will penetrate into the ground. In the ground, we have soil, means soil contains a lot of microbes, and also we found some organisms like earthworm and all be present. So they may try to degrade them chemically or biologically, or they may penetrate into the groundwater through leaching from the soil to the water. So like that, then it will slowly enter into the food, because once food is almost is like agriculture made. So this water, which groundwater which we use for the agriculture application, then it will may enter into the food like many crops like wheat, rice, and all. So there is a chance of this, but when we think of that, many times nowadays the people have been now monitoring the pesticide residues. But what about their biotransformed or chemically degraded of the metabolites of these pesticides present in the food? So that is also one of the major concern, because when we are been spraying the pesticide, it's an active molecule. Once it is entered into the plant, so through metabolism, or once it entered to the soil through the microbes in the soil, or the earthworm present in the soil, or the many other means, it may chemically degrade or chemically transform to another form, or biologically degrade or biologically transform to another form that may be called it as a metabolite of these pesticides, which majority of the times we are not been monitoring during the environmental monitoring studies. So that is one area we have to be focused in nowadays, to see into that what are the suppose many times we may say that if you monitor the pesticides in the environment samples like air, water, and soil, so we may see that many times we may observe not detected, but we are not monitoring the metabolites which may present in the soil. So that could be a scenario we have to address. Like that, pharmaceuticals, when we after using, we are throwing into the dustbin or sink, or maybe in the hospital biomedical waste. Although there are a lot of mitigation measures are there through wastewater treatment plants and all, but there may be this penetrate into the into the water, then into the agriculture lands, and then to the into the food. So these we have to understand, then only we could able to effectively mitigate them if at all they are entering, because many times the presence of large number of pharmaceuticals in the food, so food is not a good for us. So when you think of an industry scenario, now India is now going to be a big hub for the manufacturing of these agrochemicals and pharmaceuticals. If you see that this is the recent data from Ministry of Commerce, which we accessed on 10th August 2023. So we see that Indian pharmaceutical industry is a $25 billion industry now, as on today, as on August 2023. And now this agrochemical industry, now it has been growing into the $5 billion industry. So pharmaceutical and agrochemicals now been becoming a export intensive products from India to the world, and generates valuable trade surplus, and sold in more than 200 countries for pharmaceutical, and greater than 160 countries for agrochemicals. So major importers of these pharmaceuticals from India is USA, Belgium, and South Africa. And the major importers for agrochemicals from India are USA, Brazil, and Japan. In the pharmaceutical, we have been largely imported is APIs, and in agrochemicals, active ingredients like pesticide, direct technical grade pesticides are been in the bulk scale are largely imported from India, and then they will make their formulations. So we have been expecting that now the share in global pharmaceutical export is 2.3%, and we are ranked at 12th in the pharmaceutical sector, but in the agrochemical export sector, we are ranked in second. So that is the second, means we have been now almost second to one. First is in China, that is in the export of the agrochemicals to the world, and we are the second. So that means we are now been growing a lot, and we are expected that we are going to become a number one in in future. So now we can say that Indian agrochemicals nurture global agriculture to ensure food security. Indian agrochemical industry and pharmaceutical industry are the big manufacturing hubs for the world demands. So only thing that out of this we are going importing is APIs and the AIs from the in India. That we have to establish the manufacturing facilities to synthesize them. So that will make us be the world leaders very soon. So when you think of agrochemicals and pharmaceuticals, there are common issues. One is environmental impact, what we discussed in the last slides, and resistance to various organisms. Now it is becoming a growing concern that whatever we are using, whether it is agrochemical formulation, it is a pharmaceutical formulation for the need and fit for the purpose use. So what happens when we use them? So when we use continuously, so there is a chance that the bacteria, fungi, or the insect can induce resistance for those molecules. So for which it is very difficult to, when we are being spraying that, as per the instructions on the bottle, when we spray after several times of spray of the same molecule, same agrochemical onto the field, or same medicine given to the human health, after some time, we may say that after next to 3 to 4 spraying, if you spray 5th day, we may see that some of the insect may not kill. So then what the farmer will do, will give more. So that will give more resistance to the insect, more concentration. So that will give more resistance. So that resistance we have to arrest it. Then only we can able to do. So that is the reason many times nowadays now people have been saying that if you are using agrochemicals, don't use the same formulation every time. So change the formulation, that will change the composition, that will effectively kills the pest. So likewise, antimicrobial resistance is a big problem, because now antibiotics usage is growing at an alarming level in the global scenario, which we need to be arrested, because producing an antibiotic or agrochemical requires at least 10 years of effort. And also, if we are producing a new one, means we may have to spend at least 200 to 300 crores, or sometimes it may go up to 1,000 crores to develop it. Once it is induced resistance, means we cannot produce a new drug quickly, so we may have to repurpose the drugs. So that is a time-consuming process. So for which we have to make an effective strategies to arrest this resistance. That is also one of the area we have to monitor in agrochemicals and pharmaceutical sector. When you think of this agrochemicals and pharmaceuticals, where we think of a discovery and development of these two molecules, so this agrochemicals and pharmaceutical molecules, and their manufacturing, then application, whether it is in formulation form. Formulation, if you think of agrochemicals and the pharmaceutical formulation, are different strategies are there. So we have to develop them, then we have to understand them through monitoring process, and then to mitigate them. So these are the 5 areas where analytical strategies plays an important and critical role to understand, to analyze the molecule, to understand it, to understand their biotransformation, and then to understand their mitigation process developed. So that's where we have to have analytical strategies where we need to develop for the agrochemicals and product. So main steps involved is, one is the analytical strategy for the separation of the molecules and their purification. So this will exist in the manufacturing stage, or in the discovery and development stage, or in the identifying the impurity profiling. So then identification, characterization of the agrochemicals and their impurities, pharmaceuticals and their impurities in different stages of their production, manufacturing, their development, manufacturing, application, then understanding their monitoring into the environment, and then mitigation strategies. Then we have to do the quantification strategy, whether it is internal standard mode or it is a external standard mode. Then the method validation for the and verification. So if you are using a new method for any molecule to identify, characterize, and quantify, then we have to do a validation. So depending on the type of the molecule, whether it is agrochemical or it is a pharmaceutical. So there are different regulators for this. Suppose if you think of agrochemicals, we have to remember the CABRC, Central Insecticide Board Registration Committee in India. That is Ministry of under Ministry of Agriculture. So if you think of something like pharmaceuticals, we may think of Indian Pharmacopoeia Commission, then CDSCO, Central Drug Standard and Control Organization. So those teams. So if you think of in the USA, the molecules which have been, suppose if I think of agrochemicals, you have to remember the EPA, Environmental Protection Agency. So that will allows the registration of the molecule, and also they will see the environmental fate of these molecules. And if you think of pharmaceutical to be registered in US market, you have to approach the FDA, Food and Drug Administration. So likewise, there are different regulators. They have different requirements. We have to see the requirements given in their websites, and then we have to make the document accordingly for the registration of the product. So for which we may have to use the method development and validation and verification. So when we think of validation, if we are developing a new method or a method published in the journal, so that you have to do complete validation as per the suppose if it is agrochemical, then you have to follow UPA method validation guidelines. If it is a pharmaceutical, you have to follow FDA method validation guidelines. And there is for pharmaceuticals, you can have ICH guidelines, International Conference on Harmonization, ICH, which has been accepted by the almost more than 80 countries are accepting the guidelines developed by ICH, which is mutually agreeable documents being produced by this ICH. So then verification means, suppose you are using a standard method, maybe ICH method, EPA method, or FDA method, or AOAC method, something like those, depending on that. Then we have to verify the effectiveness of the infrastructure available at our laboratory for the given conditions. So that is one thing. So then impurity profiling. The impurity profiling is also becoming a major concern now. So impurity profiling, when we manufacture the agrochemical or pharmaceutical, so through the process related impurities, or something like if you have been making a biopesticides, so what are the impurities could be associated? Impurities means which is unwanted compounds, but they may pose a threat to the human health during the use of that agrochemical or a pharmaceuticals. So we have to identify this. This is a regulatory requirement, that what are the impurities associated with your process of producing the agrochemical or the pharmaceutical. Then we have to do the stability and residue studies for the agrochemicals as well as the pharmaceuticals. So how long what is the shelf life of this pharmaceutical and agrochemicals? Whether it is what is the degradation pattern? Whether it is how it is under the UV light, and how it is at the room temperature. Then how it is under the freezing and thawing cycles. So suppose you have put the molecule in the freeze and keeping the room temperature. So what could be the effect of this temperature, of the cold temperature to the room temperature to the hot temperature, hot conditions? What is the effect of these onto the molecule? Then we have to understand the pharmacotoxic kinetics, how it will distribute into the plant, and also how it will distribute into our human biological system. Then we may have to perform the forced degradation studies. So forced degradation consists of photo degradation patterns, temperature, thermal degradation patterns has to be understood, and also the shelf life So these are the forced degradation studies. Mainly they will conduct in pharmaceuticals, but nowadays in even agrochemicals also, now regulators are slowly asking about the forced degradation pattern of these agrochemicals under various stress conditions, which can induce stress to the molecule. And also this analytical strategies also plays important role to understand the occurrence, fate, and transport. Here you may have to take the matrix into the account. And then when we are developing a mitigation or remediation strategy for agrochemicals to eliminate them from the environment, so for which if you develop a strategy, so how effective it is to remove the agrochemical and pharmaceutical from the environment. So for which we need to have an effective analytical strategy to understand the effectiveness of the mitigation process. So when you think of a separation and purification, these are the two important aspects to separate the analytes in the sample or in the product and also purify them. That means it's isolation of a one single analyte in a purified, highest possible purified form. So that is the separation and purification. So we need to separate them from a mixer or separating a desired product from a mixer by removing contaminants. So what type of techniques, what we generally use analytical techniques is chromatography, electrophoresis, flotation, extraction. And for purification, we may use affinity chromatography, filtration, adsorption, or even crystallization process also to purify the compound. And the purpose is analytical separation is to identify the molecules or identify the variant. We can use analytical or preparative scale, but in purification only, we have to use the preparative phase. So the purification techniques for agrochemicals and pharmaceuticals, the purity has to be assigned to a desired molecule. So once we want to do a single molecule, we have synthesized a molecule. So once we do the synthesize the molecule, you have to remove the all maximum possible impurities from that, so that the the impurities present in that will not affect the active ingredient of that used for the as a agrochemical or a pharmaceutical. Must be able to purify sufficient amount of compound depending on the requirement. So maybe you may have to mg to gram to the even kilogram to the ton scale. We may have to design a purification strategy, and then we have to collect the collection recovery should be more than, because we may have to using many of the raw materials. We may try to recover them instead of throwing them, because we may use a large number of organic solvents. Throwing them is not a good process. So many times it's a recycling process which can use for the next batch of the manufacturing process. The equipment must be reliable, robust, and easy to use. So the purification techniques, there are three different approaches to semi-preparative scale purification. One is semi-preparative, that can purify the molecule at milligram to gram scale, and ultra fast preparative purification, and then supercritical fluid chromatography purification. When you think of a semi-preparative and preparative, the difference between these two is the loading capacity on the column, HPLC column or LC column, that, and also the ID of the column. So if you're using up to 30 mm, it is a semi-preparative. If you're using 50 mm, 100 mm columns, it's a preparative scale where we have a higher loading capacity, and we can get in a single injection, we can get in milligram to gram purifications. So this is the HPLC, basically is a semi-preparative phase. The difference between analytical HPLC and the semi-preparative is the use of the column ID and the whatever the lines flowing from the mobile phase to pump to detector. So these are the flow cell, and all these things has to be widen enough to allow the flow rate. We may put the flow rate of 30 ml per minute, maybe 50 ml per minute, sometimes even go up to preparative scale, maybe 100 ml per minute. And even nowadays, there is a dynamic axial compression HPLC columns, which is a steel columns along attached to the HPLC. We may go up to 100 mm ID, and we can produce in a single injection, we may purify more than 500 grams. So that much production affinity. Now there is industrial scale purifications, we call it as. So when you think of a purification from by liquid chromatography or even using a LCMS, if you see this diagram, so where we can have a UV, we can say with the HPLC with UV detector, we can able to purify them. And if you are using a mass can give a more good purification of the unseparated compounds. If you see the figure on the right side to you, left side to you, the right side, there is a target component A and impurity B. Those are closely overlapped peaks are eluting, which we could not able to separate them even after repeated changes in the column condition, mobile phase composition, or UV or detector. But for these compounds, we don't have any option to use other than the LCMS, that is MS signal. Even mass MS signal trigger based purification also can be done with the LC, liquid chromatography mass spectrometer. So which can able to easily purify the even the closely overlapped peaks of the chromatograph. So the semi-prepared prepared by LC is covers a wide range of compounds, and many modes are available in LC like size exclusion, ion exchange, normal phase, and reverse phase. And good chromatography performance can is good loading on the column possible because HPLC is analytical, semi-preparative, and preparative. They can three modes you can use in a single HPLC. Negative is collected fractions typically high concentration of water because mobile phase we contains a lot of water, and fractions are typically in dilute conditions. So we need to use the long evaporation time post purification. And salt form determined by the mobile phase is a big problem. When we have a salt, we cannot use MS for the purification process. So the other purification strategy is the purification by supercritical fluid. So this is one of the emerging technique in industry now, in the pharmaceutical industry as well as in the agrochemical industry. In this, we use the CO2 as a mobile phase, CO2 at a supercritical stage. Because we know that CO2 is in a gaseous phase at a normal temperature, but if we can make it a minus 70 or minus 78 degrees, it can becoming a solid. So that is a dry ice. In any organic synthesis labs, we know as the liquid, the solid carbon dioxide is the dry ice. But it when if we are putting a stage where we have a temperature temperature and the temperature above the room temperature or at a certain pressure, it can exist the properties in between the liquid and gas phase. That is called supercritical or supercritical. So at the supercritical, it has a gas here. We can fill it as a gas here, but it has a density like a liquid. So that way, this CO2 supercritical fluid chromatography has given a lot of advantage to purify the compound because of the many advantages like volatility of this mobile phase, because we don't need to use rotor evaporators to evaporate the solvents, and we can able to quickly do it, and it is having a very good, because CO2 is simply there is nothing present in that, so we don't need to purify it much also. So this is nowadays being used in large scale at the industry to develop at the kg scale purifications and all. The advantage of SFI in comparison to the preparative LC is shorter run times. If you see here, this is 1/4 of the analysis time. So if you run a HPLC of the two peaks, the run time is 24 minutes. But if you do the SFC for these two peaks, it is only 6 minutes. And we can reduce the use of the solvent. So in the same scale, if you use LC as a preparatory technique, we may have to use 12 liters of the solvent. At the same time, if you use SFC for the same purification scale, we have to use only 0.7 liters. And also this concentration, because if you are using a LC preparative purification, so we may have to use a large volume of the organic solvent. We have to evaporate it with a water. So it will take a lot of time to concentrate the. And here the so So we may, the other part is that nowadays, in even preparative SFC and preparative LC, there is a injection mode called single stacked injection. Stack injection means we know that we are purifying the two, separating the two compounds through a purification process, in which after just eluting these two peaks, we immediately inject it before the completion of the runtime of the first injection. We can inject second, then third, like that. So through the stacked injections, we may save a lot of time and also save the lot of the mobile phase. So this can give a very quick purification process of the agrochemicals and pharmaceuticals. So these advantages is the preparative SFCs covers wide range of compounds and excellent chromatographic performance it has, and faster cycle times and excellent loading on the column. So we can, because this is simply direct process where we can use it, and the collected fraction typically in higher concentration of organic with no water, less evaporation time needed, and excellent the volatile compounds, and easy to clean and maintain. So there sometimes the disadvantage is salt form determination by mobile phase, and CO2 infrastructure is needed because we have to create a supercritical stage of this fluid, for which we may have to have a lot of CO2 cylinders to be used, and then there is a infrastructure to be produced. And the other problem is that CO2 is a highly non-polar, so we may have there are some polar compounds which are non-polar compounds which is difficult to separate, for which we may add co-solvents like a stone methanol, but very small quantity of these co-solvents along with the carbon dioxide, which can induce some polarity to the mobile phase to separate the organic compounds. So once we want to do the separation done, purification done, then we have to identify and characterize and then quantify them. So for pharmaceuticals, there is ICH Q2, ICH guidelines. There are many documentation series are available in ICH. For the validate testing and validation, this Q2R1. R1 now the Q2R2 in the draft stage. So I think the document will come very soon. The final stage of the document Q2R2 for validation process. So if we are identifying, we have to establish the specificity of the method to identify the molecule. There is a plus is given. If we are testing for impurities, we may have to establish the validation parameters like accuracy, precision, even intermediate precision, then specificity, and then the detection limits also to be established. And when we are doing a quantification, then we have to establish the quantification limit and construct a linearity graph, and also we have to establish the test range to detect, identify, and quantify the compound. Then we are performing an assay with the dissolution apparatus in pharmaceuticals, and to understand the content and potency of these pharmaceuticals, we may have to conduct all the validation parameters listed in the ICH-Q2 R1 document to ensure that the method, analytical method, watch or analytical strategy used fit for purpose and need of the hour and meet the regulatory considerations. And the agrochemicals, we just have to follow the Insecticide Act 1968, which can able to illustrate the import of these molecules, manufacture, transport, distribution, storage, sale, and use. These are the regulatory requirements in which for analytical strategy is to have to active ingredient, then shelf life studies and storage and stability residue studies, where we need to use an effective strategy for the agrochemicals. So when we think of an analytical method for an assay or a related substance or impurity, enantiomer purity, chromatograph purity, so these are the validation parameters we have to establish during for the method what we have been using for the purpose. So when we think of assay, we have to establish the specificity and suitability for the identification of the compound, and establish the linearity and the test range, and the precision, repeatability, intermediate precision, reproducibility. Precision is the degree of difference between the repetitive measurements, and accuracy is degree of difference between the analyte spike and the analyte state from the instrument. And then we have to establish the solution stability and robustness. Robustness is one of the important aspects in many times. Even a small changes in the method conditions, so whether it is impacting the final measurement value of the analyte, whether it is agrochemical or a pharmaceuticals. And the pharmaceutical enantiomeric impurity, because enantiomers has now played a big role in pharmaceutical sector, because many enantiomers having a very high pharmaceutical activity. But when we have one enantiomer is highly active, but another enantiomer is highly inactive. So the purification of these two enantiomers or diastereomers or chiral molecules plays an important role. So in the activity of that molecule in the pharmaceutical sector, for which we need to establish the method which we are using to identify this enantiomer. We have to establish the specificity and system suitability. That means our method is specific enough to detect the one enantiomer in presence of the another enantiomer, or one enantiomer in presence of other diastereomers. So what is the limit of detection and quantification when we are want to this, and the linearity, calibration range, what is that calibration range, and the test rate. Then the precision, repeatability, and intercept accuracy, then solution stability. So if you are using a cleaning validation, suppose in the production scale, when we have been doing the method, when we just want to do the production, one batch is over, second batch, we want to clean all the things before that, then we have to do this, start the next batch. So we have to perform a cleaning validation of the method for which the productions, for which we have to establish specificity and system suitability, LOD and LOQ. LOD is the minimum detectable quantity of the analyte in the given sample. LOD is the minimum quantifiable quantity of the analyte in the given sample. Linearity is the calibration of construction of the construction at which we can able to detect the analyte. The calibration concentrations we have to choose. I think some noise is there. Can you mute that somewhere?
Sir, I muted, sir. Muted, already muted.
Thank you. So limit of, and then when we do the calibration, so calibration has to be, calibration range has to be fixed in such a way that when we do the unknown sample peak area has to fall in the calibration range. If it is below the calibration range or above the calibration range, either we have to dilute the sample to bring the peak area into the calibration range, or else we have to enhance the calibration range to bring the peak area of the analyte into the calibration. So that is very much important. Regulators will not accept otherwise if you don't do that. And then other part is that intermediate precision reproducibility, and these are really common. And we do the residue solvent. Once we synthesize the agrochemical and pharmaceutical, before we are going to release the product, we have to ensure that. So what is the residual solvent? Because during synthesis or manufacturing process, we use large volume of these organic solvents. Those many times are toxic, and many solvents like methanol is very toxic. It's not good for the human consumption. So then you have to establish what is the residual solvent in the final product, for which the method what we are using, maybe headspace GCMS or direct GCMS injection, for which we have to establish the validation parameters like specificity, system solubility, LOD, LOQ, linearity, that is calibration and range, precision, repeatability, intermediate precision, accuracy, and robustness. We have to build that validation document for these methods used to identify the residual solvents present in the final agrochemical or pharmaceuticals. So when revalidation and re-verification need to perform, when we change the synthesis of the drug substance, suppose we are using one route, then we want to use another route. So for which we have to identify the impurities, because once we change the synthetic route means the impurity profiling will change. In that case, we have to use we have to revalidate the method. Although we are using the same method, but we have to revalidate or re-verify the method to understand its fitness to the purpose. And a change in the composition of the drug product. If at all anything changed in the composition, maybe active ingredient used, which maybe we are just changing the percentage of the active ingredient in the formulation sample, then you have to revalidate the method or re-verify it. Change in the analytical procedure. But suppose you are using the today's thermo instrument, thermo HPLC or thermo LCMS, tomorrow you brought an Agilent LCMS, then we have to validate the method on the newly procured instrument to understand that the newly procured instrument fit for the purpose and for the need. And a major upgradation in validation verification procedure. Sorry. And a change in the manufacturing process during the life cycle of the product. So in these circumstances, you may have to revalidate the method or re-verify them before being used for the purpose. And when it comes to the understanding and identifying the compounds in the environment, for like agrochemicals and pesticide presence in the environment and food samples. So environment means water is a very simple matrix to extract the analytes from it. But when we consider the food and the soil and the biological matrix, those are very complex, because your intent may be to analyze one pesticide or one pharmaceutical product. But if you think of a food commodity, it may contain many biomolecules like fatty acids, amino acid, carbohydrate, protein, trace elements naturally available in the plants or in the food. So analyte what we are going to analyze may be not in a free form, maybe in the interacted with these food components or biological components. So we have to break that binding, then get into the free form, then analyze it. So for which we have to understand the matrix effect. So matrix effect has to be done in any sample which is very complex in nature. So we have to understand what is the interaction of this analyte with the matrix, and also how matrix is affecting the quantity of the analyte in that sample. So to understand that, we may have to perform matrix match calibration, which is the calibration means we have been taking a pure compound in different concentration, running with the method what we have been developed. But when we are That is called a need calibration. So when we want to understand the matrix effect, we spike the constant pure form of the analyte into the matrix in different concentrations and run it with the extraction, then analysis with the instrumental analysis. Then you may get calibration linearity to understand how matrix is affecting on the in analyte of our interest. So we may use to blank matrix preferably of the same type as the sample should be used to understand the matrix effect on the calibration. Matrix matched calibration is commonly used to compensate for matrix effects. So that is very much important when we do the complex samples like food, plant, and biological samples. So then after that, so these are I say validation of
ஆபீஸ்ல அந்த லேடி அவங்க கோபிங்க இருந்தாங்க, யாரோ ஒரு பையன் நிக்க வச்சு பேசினாங்க. அப்புறம் அவங்க அதை அனுப்பிட்டாங்க. ஹெச்ஓடி தான் மாமா. வேற வேணும் இல்லை இவங்க உங்க இதுல இருந்து மினிட் டு மினிட் அனாலிஸ்டி. இல்லை ரேட் பாருங்க.
Pramod sir, please unmute Mohan sir. Pramod sir, please unmute Mohan sir.
அங்கவே ஏதோ ப்ராப்ளம்.
Pramod sir. Pramod sir.
Mohana Krishna Reddy is presenting.
हाँ सर. हाँ मेरा तो ये चल रहा है, कॉल चल रहा है यार, यहाँ वेबिनार चल रहा है.
मोहन सर जो स्पीकर है उनको अनम्यूट करिए ना सर. वो नहीं कर पा रहा है, बराबर कर से जाएगा.
Mohan sir, you can also unmute yourself, sir. You are now co-host. Hello.
Yeah. So are you able to hear me?
Yes. Now. Yes, sir. Able to hear you.
Yeah. Okay. So then comes to the quality control and quality assurance. These are the two important parameters when we are working in industry to understand that what would be the quality control aspect. Quality assurance is the proactive approach of quality which focuses on preventing the defects at the process level. And quality control is the reactive approach of quality which works by finding the defects of the product. When we are able to see that why QA is different from QC, whereas QA is focused on the manufacturing process. The process involves the preparation of patient and establishment of the organogram, and also seeing the how QC department is working. All this comes under quality assurance. But the quality control is the quality inspection of the product at the manufacturing stage. to evaluate if they pass certain quality standards before being shipped to the customers. QC aims to catch deficiencies in quality, while QA aims to prevent deficiencies from occurring. So QA is a preventing process, and QC is a detective process. So that is the true. QA is part of the QC is part of the quality control, is part of the quality assurance. So when you think of the QA and QC, so we have two certifying and accreditation agencies in India is available. One is NABL, sorry, National Accreditation Board for Testing and Calibration of Laboratories, and other one is NGCMA, National GLP Compliance Monitoring Authority, which certifies our laboratory whether it is compliance to GLP compliance to OECD principles. OECD means Organization of Economic Cooperation Development. That is been adopted by the 80 countries, which will develop documents to understand how good laboratory practices to be developed in any laboratory, which is involved to do the preclinical safety study, preclinical health research, preclinical studies, as well as environmental safety studies. So OECD GLP is very specific, and ISO IEC also. So when we think of the overlapping subjects of these two accreditation and certifying agency, is one is the management, which is common for both. The only thing that NABL gives some quality management structure. They will ask management structure, but GLP and NGCM ask for the organogram. So that is then training, reference materials use, equipment and maintenance, report preparation, sampling during the study, sample reception, audits, corrective action, method validation, quality control procedures. These are common in both the things. But what is the specific things are in GLP, OECD GLP is, one is master schedule, then the study director. Study director is the whole soul responsible for the study being conducted by the laboratory. Suppose you have been asked to do a toxicity study of the new formulation. In that, then the test facility management in GLP certified facility will assign that study to one study director. So that study director will be the responsible to conduct, to plan, execute, design, conduct, complete, and report to the client. So he is the whole sole responsibility. Then archivist, there is an archivist has to be there. Once the study has been done, before sending the report to the client, the study report has to be archived at the department. That will be available for at least 10 years if any problem comes with the product under study. Then study plan has to be made. So any study has to be conducted in GLP certified facility, has to have a study plan which has to be approved by the test facility management as well as the sponsor. Sponsor means the client who has given the study to you. Then the test article and test system. Test article means it's a test item. So the test item is the product received to perform the studies on which. Test system is, suppose you are studying the product in animal, that is the test system. So you are analyzing that test item in the instrument, that is the test system. And what is the chain of custody of this whole system? And then inspection monitoring. So we will have to have a quality assurance unit, which is the heart of the any GLP certified facility. So they will monitor the facility inspections as well as the study inspections. They will identify the critical quality attributes to be inspected during the study under progress. So study director has to inform to the quality assurance to do the before initiating the study, and even study plan also to be verified and reviewed by the quality assurance, without which the client or TFM cannot approve it. Likewise, the survey, when it comes to the ISO IEC 17025, this is the standard guidelines followed by the NABL to evaluate the competency of the testing and calibration activity of the any laboratory, in which there is a compliance section. Then we have the uncertainty of measurement. So measurement uncertainty is an important aspect in any test method what we have been using to understand, because when we think of whatever the value we are reporting, it's an experimental value, but we don't know what is the actual value of that analyte in the sample. So to understand whether that process of measurement to identify or quantify the analyte in the sample is close to the true value, because true value we cannot able to measure. So that so for which we have to understand to define a range in the system in which we may expect that true value can fall. So for which to identify the measurement of the uncertainty, we have to identify the sources of uncertainty. Suppose you are weighing a compound in the balance. So what is the uncertainty of the value given by the balance with respect to the weight we have been done with that balance? So when we calibrate the analytical balance, the calibration agency will give the measurement uncertainty value for that balance depending on that their procedure. So we have to use. Then when we do the calibration of the calibration graph is constructed for the analyte in the sample analysis, then we have to define the what is the uncertainty induced by this calibration to quantify the analyte using that graph. So this is an important parameter has been given a separate clause in ISO 17025 2017 standard. So we have to understand that is very specific to enable activity. Then proficiency testing, the laboratory has to participate in the proficiency testing to establish the competency in the identified scope. Then we have to do the preventive actions if anything falls or deviation from the existing system. Then services to the client. So these are very specific to enable accredited labs. So this has to be followed by any testing or testing laboratory which performs enable testing or a GLP study. So when we see that ISO 17025, which enables laboratories to demonstrate that they operate competently. So competency of the laboratory has to be established as per the guidelines given, as per the clauses given in ISO IEC 17025, and generate valid results, thereby promoting confidence in their work both nationally and around the world. It helps to facilitate cooperation between laboratories and other bodies by generating wider acceptance of results between the countries. So once your laboratory is accredited with NABL as per ISO IEC 17025, then your data is accepted at globally. So the client who sent the sample to you need not to retest the sample in the product. Has, suppose if the product is exported to Europe, so the Europe regulatory authorities, once they see that your certificate of analysis is has enable accreditation, then they will not retest the sample. So that is the global acceptance of the results produced by the laboratory if you have enable accreditation, or like a GLP, even GLP also same. The test reports and certificates can be accepted from one country to another without the need for further testing, which in turn improves international trade. So that also, so what are the benefits of having accreditation is that once you got the laboratory get national and international recognition, and it reduce cost of retesting, and also better control over laboratory operations, because you have a systematic procedure which has been completely documented in every stage of the testing activity. So that way you can understand that where we have been going wrong. Suppose if any result gone wrong, so you can easily identify that where in which step or which stage we have been gone wrong, so that we can do some corrective action to prevent further future testing activities, and to prevent errors in the future testing activities, and increase confidence in testing and calibrations. And also we have a because once we are enable accredited means many industries will approach you to do their product testing. And validity of test methods and provision of the reliable data is assured if we have a accreditation as per ISO 17025. So this is a big document. So I think just overview that once we want to go to enable accreditation, we have to have general requirements to impartiality and confidentiality of the documents or work being performed by the laboratory, and the structural requirements. There is a managerial aspects, the laboratory origin of the laboratory, what is the evidence for the origin of the laboratory, and what is the structure of the management structure of the laboratory has to be addressed as per the clauses in 5 structural requirements. Then we have to establish the resource requirements like general. In general means total infrastructure. Personal means the personal competency, training, test, then their qualification and all to be addressed as per these clauses. Then facilities and environmental conditions has to be recorded, monitored, and also the equipment has to met as per the clauses in 6.4 of ISO IEC 17025, and to establish the metallurgical traceability of the equipments to the SI system. That is very important. And also externally provided products like process requirements is to review, request, tender contracts, and then selection, verification, validation of the methods, then sampling aspect and handling of test and calibration items, then technical records and evaluation of measurement. So the separate report has been given, the evaluation of measurement uncertainty, reporting of results and non-conforming work. So these are the management system requirements also as per the clause 8 has to be addressed by the laboratory to establish and evaluated by the NAPL. So when we think of a GLP certified or GLP compliant facility, so you have to understand the principles of GLT. Start with the testing facility, organization and professional, quality assurance programs, apparatus and material reagents, test systems has to be established. If it is animal, then you have to build animal facility. If it is something like a microorganisms, you have to build that microorganism facility as per OECD document series. There are the OECD document series are available. You can just simply get it from the Google. There's an open access documents. And test and reference substances and standard operating procedures. You have to prepare the documentation, how we will receive the sample, what is the procedure to transport the sample from the receiving to the laboratory, then how they will distribute the work, like all these things. We have to build a SOPs. Then the facilities and the performance of the study evaluated by the quality assurance unit, and the reporting of study results, which has to be accepted by the client. Final report has to be accepted by the client. Then only that is the process is completed. Then storage and retention of records and materials, that is archiving process. So these are the steps you have to build it in the facility to get it GLP compliant or a GLP certified by the National GLP Compliance Monitoring Authority, which is under the Department of Science and Technology. So this is my overall talk, and thank you. Any questions? You're most welcome.
Thank you, sir. Such an informative session, and I think you all participants are enjoyed for this session. Is there any queries from your side, the participant students of agriculture and pharmacy students, forensic students? If there is any questions, please ask to the sir. One by one, please. If you don't have question, I received some questions from my personal number. So can I ask, sir?
Yeah, please go ahead.
Okay. So just a minute, sir. One question is there, sir. What are the advantages of natural agrochemicals or pesticide over the other synthetic pesticide or herbicides? This is the one question I received from the one participants.
Yeah. So natural agrochemicals is easy to prepare. One is the process is very easy to prepare. So the second one is their biodegradability and bioavailability is very high, and it has a broad spectrum of insecticidal properties, because we may have to use a single extract to kill many pests, different varieties of pests. So these are the major advantages what we see in the natural bio-based pesticides.
Yes, sir. So how do the agrochemical affect the environment, sir? One more
So if
question
you
is
think of agrochemicals, synthetic agrochemicals like pesticides, suppose if you see organochlorine like DDT, endosulfan, and gamma HCH. These have been widely used in the last 10 years back, and all maybe last 100 years people have been using, and they're highly persistent, and over the time they will not degrade from the environment. So they're becoming a big threat to the humans because they are highly toxic. So that is the only pattern. So for which now there are a lot of replacement of organochlorine with organophosphorus with good half-life, and then followed by pyrethroids. Now herbicides are coming. So they have become present in the environment, and finally it is enters into the food, and we have been consuming them through food. So that is not good. Yeah,
okay,
please.
sir. So is there any specified procedure for the disposal of pesticides, agrochemicals?
Yeah, specified procedure means basically we have to, suppose if it has been present in the environment, then we have to mitigate it with some bacterial degradation. So we may have to isolate the bacteria from soil. We have to use it in the larger scale to mitigate them. Or mainly means mitigation means it's a conversion of this pesticide into metabolite, which might be less toxic than the pesticide. So that is the one mitigation process. Otherwise, the manufacturers will do their effluent treatment plants are been done, effluent treatment procedures are been available to ensure that those will not enter into the environment during the manufacturing process.
And, one question, sir, more, sir. Can we classify the pest or the same pesticides are used in all seasons, all throughout the year, like seasonal pest and any other pesticides?
Yeah, pesticide classification is broad, is there with many type of classes are available, but the use, when you think of use, what class of pesticide to be used, that purely depends on the type of the crop. The type of the crop and also based on the usage and agroclimatic conditions of that region where we are going to use the pesticide.
Okay, sir. You have discussed in your presentation, sir, is the estimation by the liquid chromatography of pesticide and agrochemicals or pharmaceuticals. So there is one question, which type of column we can use the frequently for the method development as the agrochemical uh, and natural product are getting problem for the column and all. Are there pre-analytical column we are using in HPLC? So is there any specific column we can use for the estimation of method development of pesticides or
Yeah,
agrochemicals?
one is that suppose pesticides mainly of hydrophilic in nature, many of the pesticides, we can use gas chromatography mass spectrometry instrument with one column most widely used is 5% phenyl, 95% methyl siloxane. That is
Yes.
something like DB5MS, like that, Elite 5MS based on the manufacturer. We can use. So most of the pesticides can be analyzed with that one. So if any the pesticides are hydrophobic, so hydrophilic. So lipophilic means you have to use GCMS. Hydrophilic means you have to use HPLC. Many of the pesticides can easily be analyzed by C18 or C8 column.
Okay, that means the hybrid technology of method development like LCMS and
Yeah.
we can use.
Suppose
Yes, yes.
many of the 300 pesticides can be analyzed by GC or GCMS
Hello.
with
Yes, sir.
5% phenyl, 95% methyl siloxane column.
Sir, actually, I have a small doubt. Actually, it is not related to your topic. Now, biofertilizers, biopesticides, they are using very popularly in our country. So, these are actually required permissions or not required, sir?
Permission means as such now there is no complete guidelines, sir. But now I think CABRC is bringing some guidelines like biopesticide and biostimulants. They're bringing some regulatory things, but not in the final place to follow.
Okay.
So
Till then, they can go. Till then, without permission, they can go like this. As
without
this.
permission means we have to go for the registration. The minimum guidelines is very minimal in comparison to the synthetic pesticides.
Okay.
So we have to go
And,
CABRC registration for even for
Yes, sir.
biopesticides.
Yes, sir. Yes, sir.
okay, sir.
Thank you
One
very
more
much, sir.
question.
Actually, you are given a very good information. Questions can be continued, I says.
Okay, sir. Sir, one more question is there. What is the actual role of NABL for the
Yeah. NABL is basically it's
pharmaceutical?
accreditation body in India because sorry, I think
Exact
can you
role.
repeat the question? It was the voice is broken.
Actually, what is the development and agrochemical development industries? Is there any requirement for any annual accreditation certification for the production of agrochemical and pharmaceuticals?
Yeah. So enable is a accreditation body in India because we have a international metrology organization in France, Paris. That is the primary metrology body, and each country has one NMI, National Metrology Institute. So in India, we have a National Physical Laboratory is our NMI in India. So enable is they follow some standard guidelines. It's a third party to accredit the and evaluate the competency of the laboratory as per the ISO 17025 guidelines. So if any laboratory wants to get accreditation from NABL as per ISO IEC 17025 and they place the application to them, they will not involve in any testing activity. NABL is just an accreditation body to just evaluate through their team of assessors. So they will come, they will send an assessors team. Once you file the application to them, they will come, they will send an assessors, there is technical assessor and lead assessor. They will come and see your laboratory infrastructure as per ISO IEC 17025 clauses. And then if any non-conformances are there, they will raise it and submit to NABL. Then laboratory has to address those non-conformances to the NABL. Once non-conformance addressed, then NABL recommends the accreditation of the laboratory in the testing and calibration activity only. Suppose in agrochemicals, you have
Yes.
to test the molecule with respect to active ingredient, analytical test report, and residue studies. So where we can go for the enable accreditation.
Yes, sir, and very much thank you, sir. First of all,
So there is a
I thank respected. Uh,
yeah, there is one question
this is
from
the
yeah, why
yes, sir.
not use more and more natural fertilizers rather than synthetic fertilizers for minimize environmental pollution? So which can be done because now there is a, now we have been completely transforming to the bio-based pesticides and biofertilizers now. So, but the process are involved little tediousness of these biofertilizers. So, which the many of the now industries are addressing them. So, slowly we will completely we may go to the biofertilizers. Yeah. Thank you.
Is there any questions from participant side? Otherwise, I will go for vote of thanks first, sir. Okay, I think there is no question, sir. So, in this session, first of all, I thank respected VC sir, Professor K.R.S. Sambasivarao sir, and Pro VC ma'am Vinita Saluja ma'am, who inspired us and encouraging to conduct this webinar and made us take it further. After that, we are grateful and would like to heartily thanks to our today guest and speaker, respected Dr. Mohana Krishna Reddy, Mudiam sir, is a director, Institute of Pesticide Formulation Technology, IIT, who gave a fantastic and informative session and address on agrochemical and pharmaceutical analytical strategy and regulation aspects. Thank you very much, sir. Thank you to all the students of Pharmacy and Agriculture Department of Mangalayatan University and students who participate outside the university for the attentive during the webinar very well. Last but not least, at the end of webinar, I would like to thank everyone who's joined the webinar, my colleague, academician, and dear students again. And I also request to today's speakers, respected Professor Dr. Mohana Krishna Reddy, Mudiam sir, to continue giving such wonderful and informative webinar in future, sir. And dhanyavad, sir.
Thank you, Rajesh. Okay, bye. Thank you.
Is there? Okay, sir. Thank you to all of you. VC sir, I think he is engaged in inspection, sir. So we are going to end this session here only. VC sir is there. Respected VC sir. Okay, I think is not in. Okay, sir. Thank you again, sir. Mohana Krishna Reddy, sir. I will contact you on your mobile, sir. If there any doubt, I will contact you, sir. And I think everybody enjoyed this session because Mohana Reddy is a very dynamic personality, as he is given very informative session to all of us. Thank you, sir. Again.
Thank you, Rajesh. Thank you all. Bye.
Okay.